The immune response has 4 basic steps. First step would be to detect and identify the foreign substance. Number 2, communicate with other immune system cells, and then recruit assistance and coordination of the response. And the 4th step would be destroy or suppress the invader. So let's take a look at the lymphoid tissues. Pretty much throughout the entire body, but the primary lymphoid tissues would be the thymus gland and bone marrow. Secondary lymphoid tissues include spleen and lymph nodes, and then we have some diffuse lymphoid tissues like tonsils, the gut-associated lymphoid tissue, and clusters of other lymphoid tissues. So here will be the anatomy summary of the lymphatic system with lymph nodes, and we can just review that. Here are the cells that are involved with the immune responses. So previously in the blood lecture, of course, we focused mostly on the red blood cells and some other blood-related items, but we also already took a look at the white blood cells. And here, just for review, we have granulocytes and agranulocytes. And so here, the types of cells that are listed right here, basophils and eosinophils. These are rare types of cells. The first 3 here, together with the neutrophils, those are your granulocytes. Okay, so here, granulocytes, the first from here to here, granulocytes. So there will be basophils. We have very few of them in circulation because they almost immediately leave your blood and migrate into tissues where they're called mast cells. And you do need to know both of those terms. Basophils in circulation are mast cells once they're in tissues. The granules are filled with histamine, and they are kind of responsible for your inflammatory responses, also for allergic reactions. Neutrophils, that will be your most numerous type of white blood cell overall. Here we have 50 to 70% of your white blood cells in circulation are neutrophils. They have that odd-shaped nucleus, so they're called polymorphonuclear neutrophils. And the nucleus has like 3 to 5 lobes. The granules are generally not visible because you don't offer them a neutral dye, so they don't show up with the acidic or the basic kind of dye that we use for the staining of the eosinophils and the basophils. So the neutrophils, that is maybe the biggest part of your second line of defense, because the neutrophils, you have so many of them, and they are attracted to sites of inflammation, and then they're just phagocytic. They're highly phagocytic cells. Eosinophils are rather rare too. About 1 or 2, here says 1 to 3% of your white blood cells in circulation are eosinophils. They're granules stained with that orange dye, the eosin, and they will be mounting a response against eukaryotic parasitic type of infection. So if you have pinworm infection, your eosinophils will be brave enough to go after those. Another important, so here we're moving on to the agranulocytes. The first one here will be the monocyte. That's what they're called when they're in circulation. Once a monocyte migrates into tissues, it is called a macrophage. Macrophage meaning big eater. Together with the neutrophils, the macrophages will engulf particles and just do a lot of phagocytosis. Lymphocytes, they will differentiate into B cells and T cells. I'll write this up here again, B cells. And they, of course, make antibodies. And then T cells, therefore cell-mediated immune functions. Plasma cells, those are cells that are B cells that are actively secreting antibody. When you don't need that antibody anymore, then these plasma cells, they will keep a sample of themselves around, and these are called memory cells. We'll get that later. But lymphocytes in general, they will be taking care of your specific immune functions, and they can differentiate into B cells that make antibodies and T cells for cell-mediated functions. And then here one last cell, dendritic cells, they're also for specific type of immune functions. Now let's take a look at how a phagocytic cell might engulf and then destroy a pathogen. So here's our pathogen, a bacterium apparently, and here's our phagocytic cell. So that could be your neutrophil or your macrophage. It will approach this bacterium, it will engulf it, so it will form these pseudopods, and then it will recognize some sort of a surface structure here on this pathogen, and then keep engulfing the bacterium until it's fully internalized. It will fuse it with a lysozyme and then pick this bacterium apart. Here we have another version of this scenario. Here in this case, we have a bacterium with a capsule. Capsule is kind of like a coating that's invisible to the immune system. And so anytime a bacterium has a capsule, it's kind of like a stealth fighter. It's really escapes the radar of the phagocytic cell, and so you need to first coat that bacterium that has a capsule. You need to coat that with antibodies so that these antibodies then guide the phagocytic cell. Oh, okay, this is what I should engulf and eat up. So you need to first have antibodies that are coating this bacterium, and once it's been coated, then it's called opsonization, and this way this macrophage or neutrophil phagocytic cell can engulf then the bacterium, internalize it, and pick it apart. And so here would be the continuation. So now we have this bacterium has been internalized, and now we need to fuse it with the lysosome, and the lysosome contains, of course, the hydrolytic enzymes. And so once you fuse that, now you're picking apart this bacterium, and all here, what's in here is basically just picked apart pieces from this bacterium. Now what macrophages really like to do is, once they have this phagolysosome right here, and they have picked apart this antigen, so a digested antigen. So now they're taking characteristic pieces from the surface of that bacterium and show it to the rest of the immune system, and then in this way this phagocytic cell has now become an antigen presenting cell, an APC, antigen presenting cell. And so basically this phagocytic cell says, oh, okay, this red triangle here is super interesting, so I'm going to present that to the rest of the immune system and see what they think about it. So you will take a self-recognition complex, an MHC complex, and sort of that's the box right here, and then embedded into the box is the red triangle. And the red triangle came from the bacterium, and your phagocytic cell thought it was very interesting, so it's now displaying that red triangle as sort of a feature of the enemy to the rest of the immune system. So now we're going to move on to innate immunity, and that's of course part of your non-specific defenses. And basically innate immunity means that you're born with it. The first thing that comes to mind, of course, are your physical barriers such as unbroken skin, and then also chemical barriers. Here the example is given, lysozyme. Lysozyme is an enzyme that's found in the saliva in your mouth, and it breaks down bacterial cell walls. Now, phagocytes, they belong to your second line of defense. So they eat foreign material. They could also eat cells that are damaged, for example, but they're specializing in eating up bacteria, for example. Now, one of the ways that you can actually direct a phagocytic cell toward a site of inflammation or where some infection is going on, the easiest way is by chemotaxins. Chemotaxins are chemicals that direct the phagocytes, these white blood cells that are phagocytic, to come to an area of inflammation or infection, and then eat as many bacteria as possible and try to prevent the spreading of the infection. Once these phagocytic cells are filled up with bacteria, they die. So they die in battle. They have eaten as many of the enemy cells as they could, and then they just died. And dead neutrophils, basically, that's what we call pus. Then we have toll-like receptors Those are basically surface receptors that will bind to pathogens and then sort of start a cascade of events telling the immune system what it should do or what it could activate. So it's a group of receptors that can interact with pathogens and then communicate sort of signal transduction pathways that communicate into the interior of those white blood cells. Opsonins, those are basically also coding mechanisms where you can attract, let's say, a macrophage, make it easier for the macrophage to detect a bacterium that it should eat. So it's kind of, chemotoxins are more released to attract white blood cells, and opsonins, those are more substances that signal to a white blood cell, hey, you need to eat this thing. Now moving on to natural killer cells. These guys here, those are cells that kill infected cells. For example, virus-infected cells. They behave abnormally, and natural killer cells, they can detect that and kill virus-infected cells. And they also, their job is to find cancer cells, tumor cells that are behaving abnormally, and they will attack them and kill those kind of cells. Cytokines, it's a whole group of molecules that communicate between white blood cells. They can also help with the inflammatory responses. So basically, they're communication molecules in the immune system. Here a little bit more information on NK cells, the natural killer cells. They kill infected, virus-infected cells, that typically is, and tumor cells. So these natural killer cells, they recognize virus-infected cells and then induce apoptosis, so to prevent spreading of the virus particles. And natural killer cells, they also attack some tumor cells. It's nice to have sort of your defense against, cancer cells. Before they can cause a problem, your natural killer cells, they're supposed to recognize those abnormal cells and kill them. Natural killer cells and other lymphocytes, they also secrete antiviral cytokines, including interferons. Now, interferons is sort of your natural virus defense. Basically, interferons, that's a warning mechanism for cells that have not been infected that viral attack is imminent. And so basically, there are different types of interferons. They're actually available via biotechnology these days, but they work best right there locally when they're secreted by natural killer cells, for example. So here, different types of interferons here: alpha interferon, beta interferon, so that they prevent viral replication, and gamma interferons may be the most widely used, or the one that you may have heard about the most. And they can activate macrophages and other immune system cells. Cytokines, they create inflammatory responses. They also communicate between white blood cells. And let's talk about inflammation. Inflammation is a hallmark reaction of innate immunity. And it is so severe that in the old days, the Greeks, they thought it was a disease. So inflammation was described in one of the earliest medical papers that are available. And basically, the Greeks, they described inflammation as a disease. Now, the important roles here of inflammation, well, we don't like it usually, but inflammation attracts immune system cells and chemical mediators to the site, so they're calling a whole army of neutrophils and macrophages over to the area where those cells are needed. They also produce physical barriers to slow down the spreading of an infection and sort of limit it to a local area. And they also promote tissue repair once the infection is under control. Now let's take a look at, I'm just going to give you the hallmark steps of inflammation as written in that early medical paper. Let's see where we have some space. So here the old Greeks, they were saying that the 4 cardinal signs of inflammation are, number 1, rubor. Rubor means redness. You know this, when a site is inflamed and it's red. Number 2, dolor. Those of you who speak Spanish, you know what that means. Dolor is pain. And number 3 is tumor. Tumor means swelling. And number 4, we're going to have calor. And you probably know what that means too. Calor is heat. So also the term calorie, we know that's an amount of heat. Anyway, so these are the 4 cardinal signs of inflammation as written in this earliest paper of in medicine by the Greeks, and they use these terms to describe inflammation as rubor, the redness, dolor, the pain, tumor, the swelling, and calor, the heat that is associated with inflammatory responses. So here I wrote this out, 4 cardinal signs of inflammation.